Hello friends, welcome to Simply Biology. Today we discuss about the different types of DNA and the alternative conformations of bDNA. The DNA is classified in various ways on the basis on the number of strands, based on the number of nucleotide residues, based on the shape of DNA, and based on the nature of nucleotide sequence in Based on the number of strands, the DNA is classified into two types. They are single-stranded and double-stranded. And another classification is based on the number of nucleotide residues in DNA molecule. They are aDNA, bDNA, zDNA and RL helix model of DNA. Based on the shape of DNA, DNA is classified into three types. They are circular DNA, relaxed DNA and supercoiled DNA. And the last classification is based on the nature of nucleotide sequence in duplex DNA. They are palindromic DNA and repetitive or satellite DNA. Let us study in detail all these types of DNA. On the basis of the number of strands, DNA is classified into two types, namely double-stranded DNA and single-stranded DNA. DNA of some viruses such as Psi-174 infecting E. coli is single-stranded. The double-stranded DNA is otherwise known as double helical DNA. In most of the organisms, except a few viruses, the DNA has a double-stranded structure. On the basis of the shape of DNA, they are classified into three types, namely circular DNA, relaxed DNA and supercoiled DNA. Strand. It is found in some viruses like Psi-174. Double-stranded circular DNA is found in bacteria, most of the viruses, mitochondria, chloroplast, etc. The circular DNA without any helical coiling is called the relaxed DNA. In supercoiled DNA, the axis of the double helix itself is twisted to form a superhelix. It can produce negative supercoiling and positive supercoiling. The degree of coiling is controlled by topoisomerosis or gyrases. On the basis of the nature of nucleotide sequence in duplex DNA, they are of two types, palindromic DNA and repetitive DNA or satellite DNA. Palindrome is a word or sentence that reads the same both forwards and backwards. Palindromic DNA is a DNA containing palindromic sequences of nucleotides. In palindromic sequence, the sequence of nucleotides goes in one direction in one strand and in another direction in the second strand. It is an inverted sequence. The term palindromic DNA was first used by Wilson and Thomas in 1974. The palindromic sequences are found in the DNA of both prokaryotes and eukaryotes. The palindromic sequences may have a length of 3 to 10 nucleotides or hundreds or thousands of nucleotides. Comparatively, the palindromic sequences of eukaryotes are longer than that of prokaryotes. When very short sequences of base pairs are repeated many times in DNA, the DNA is called repetitive DNA or satellite DNA. In repetitive DNA, Some regions are repeated in 100 or 1000 times. This is a linear DNA, the telomere, the centromere and the telomere of a chromosome. These blue regions are the repetitive DNA components, and the space in between two repetitive DNA components is called the spacer region. All eukaryotes except yeast containing repetitive DNA. Repetitive DNA is absent from prokaryotes. In human, 30% of the DNA contains sequences which are repeated at least 20 times. The double helix structure of DNA is occurred in 6 different forms: aDNA, bDNA, cDNA, ddNA, eDNA and zDNA. Among these, aDNA, bDNA and zDNA are important forms. bDNA is the Watson and Crick double helix having 10 residues per turn. It is a right-handed DNA. aDNA is discovered by Rosalind Franklin. It is a double helical DNA having 11 residues per turn. It is a right-handed helix. It is formed by the dehydration of bDNA. Double-stranded RNA also exists in A form. It has 11 base pairs per turn. The sugar phosphate backbone is regular. The repeating units are mononucleotides. The ecDNA is first discovered by Robert Wells and his colleagues. It is a left-handed double helix having 12 base pairs per turn. The sugar phosphate backbone is zigzag in orientation. The ecDNA has some alternating repeated units of dinucleotides throughout the DNA. The ecDNA model was proposed by Rich. It plays a role in genetic recombination. Look at this picture. The aDNA is shorter and broadest than bDNA, and the ecDNA is long and thin as compared to bDNA. Here you can see the DNA backbone in ecDNA were in a zigzag manner. The reason behind this is, in ecDNA, the purine nitrogenous bases and the pyrimidine nitrogenous bases are in different conformation. The purines shows a syn conformation. That means the bases and sugar are near and on the same side, while in pyrimidines they are in anti-conformation. That means the base and the sugar are distant and on the opposite side. According to Rotley and Shashishekaran, the DNA duplex is formed of alternating right and left-handed helices arranged side by side. Hence, this model is called right and left helix model of, or RL helix model of DNA. Each strand has 5 base pairs in the right-handed helix, alternating with 5 base pairs in the left-handed helix. This model has some advantages such as, during replication, the DNA duplex unwinds without twisting around each other. And the second advantage is, this model helps in the supercoiling of eukaryotic chromosomes without the presence of King's irregular helix. Here you can see a table which shows the difference between aDNA, bDNA and zDNA. You all know that the bDNA is the DNA model proposed by Watson and Crick, and the aDNA and zDNA are its alternative conformations. The helix type of aDNA is right, bDNA is right, and zDNA is a left-handed helix. The bDNA is normal and it has a diameter of 2.37 nanometer or approximately 20 angstrom, and the diameter of aDNA is 2.55 nanometer and its of zDNA is 1.84 nanometer. The distance for each complete turn in bDNA is 3.4 nanometer, and that of aDNA and zDNA is 3.2 and 4.5 respectively. The rise per base pair in bDNA is 0.34 nanometer. This is the distance between two adjacent base pair in a single strand, and that of aDNA is 0.29 and that of zDNA is 0.37 nanometer. The number of base pairs per complete turn in bDNA is 10 and in aDNA is 11 and in zDNA is 12. The base pair tilt in bDNA is 19 degree and that of aDNA is minus 1.2 degree and that of zDNA is minus 9 degree. bDNA has a wide and quite deep major groove and aDNA has a narrow and deep major groove. The bDNA has a narrow and deep minor groove, whereas aDNA has a very broad and shallow minor groove, and the zDNA has only a minor groove and that is very narrow and deep. If you like this video, please like, share, comment this video and subscribe the channel for getting notification for the upcoming videos. Thank you.